Thank you very much, Philip, for agreeing to give a Build a Cell seminar. And we can get started.
Thank you very much for the invitation. So don't hesitate to ask questions. I think that's the spirit, even in the middle of my presentation. I work at ESPCI, so it's a school of physics and chemistry. It's an engineering school initially. That there are labs, and it's located in Paris, and it's part of the new University Paris Sciences Let, so PSL if you never heard about it. And so I'm a soft matter physicist by training, and I moved to quantitative approaches to evolution, and ended up working on the origin of life, because also along my path, microfluidics was an important component. And so the study of compartments is quite central in what I will show here. What compartments can do, like how do they help? And yeah, so I ended up working on origin of life, doing experiments, which brings me close to the concept of build a cell somehow. And I'm going to talk about how some Darwinian properties could be implemented and possibly combined in physical-chemical systems. So, yeah, the question that many of you know is how do you go from abiotic Earth to some process of cells that grow and divide? And it's clearly a question. I mean, it was not a clear question for everyone until Pasteur showed that there was not spontaneous generation. At least we can say that from that time, it's quite clear. So still, it's a unusual question, I would say, from if you take it from a very disciplinary angle, chemistry, physics, or other fields, because we have in mind the focus to a historical event, and this event has a quite uncertain context. But the main big problem is that origin of life is not something that we observe really happening all the time, and that was a demonstration of Pasteur. So it makes it particularly difficult to study, in the sense that what we have to do first is create an instance or some instances of origin of life or part of it, so that we can study it and at the same time understand them. And this is why the synthetic biology, biochemistry approach is very central now. The other problem is that, unlike for other evolutionary problems, I would say maybe the origin of multicellularity or other major transitions, origin of eukaryotes, you can expect some backward inference from the tree of life. But the origin of life is really so remote that going before Luca is really hard. So we have little clues also coming from biology. Now biology is giving us a lot of examples of things that work. So biology is, you could say, is at least an inspiration. I wanted to show this. It's a work we did with Christophe Malater, who is a philosopher of science. And it arises from, well, when I go to Origin of Life conferences, there are typically misunderstandings, but there are a lot about what people expect from the answer. And so that's why we wrote this title, The Origin of Life, What is the Question? And so we separated the criteria along 3 axes. Is it historically adequate? Do we look for making life which looks like life as we know it, so similarity with life as we know it? And is the process natural, naturally spontaneous, in the sense of spontaneous? So when we think about first the origin of life question, we think about all those criteria together. How did life appear on Earth and the life that we know? But I generally take to relax any possible criterion here, because we already don't know how to answer the simpler question where none of that is true. And I think it's interesting also to relate that to the question of artificial cells. So what do we learn from artificial cells? It's not completely clear. I mean, we think it's something useful to understand the origin of life. So somehow it participates to this decomposition of the criteria. When we do an artificial cell, a priori, we put a lot of emphasis with life as we know it, and we don't care about historical adequacy or spontaneity. And of course, what we argue in this article is that any approach is helpful. So if you make a completely synthetic system, then you can think about, can you naturalize this system in terms of appearance? Can you imagine a scenario of appearance of this system? So at the moment, we know so little that every contribution is valid, I think, in any of these corners. So we have this natural aspect, and this leads this problem of historicity and lack of observation in the extant world leads people to think about scenarios. So scenarios are of course, very general in research, but in original life, they are completely central. You must go through this exercise of positing some steps and trying to see if they are valid and if there can be a chain transitions between those different steps. So a very usual way to think, and nothing original here, to think that at some point you have prebiotic chemistry leading to building blocks, which is a big problem in itself. At some point, they can oligomerize into some polymers or pre-biopolymers, which is also a problem in itself. And those are problems I don't study. I start from the fact that there are probably some oligomers. Now, what can happen is that we know they can fold. And when they fold, sometimes they can acquire some catalytic properties. And in particular, if they help their own formation, you can have an autocatalytic set. But an autocatalytic system, which we will see, is not evolving a priori. I mean, it's not obvious how it could evolve. And what we have in mind for evolution is template-based replication. So there is a sequence, the sequence is being copied, and we know you have mutations and you have some heredity because there is a copying. And by some ways, the sequence, they may fold, they may be expressed in cells and lead to some phenotype and selection. So at least from this point, it's very acceptable that you can have a form of evolution, but this requires very sophisticated objects somehow. At the moment, it's still a complete mystery whether we are progressing, but there is no satisfying accepted answer. And yeah, I draw compartments, but the compartments, they appear at the end here before Luca, but maybe they were there before. I think that's kind of the conclusion of the talk. I'm going to list arguments for which go in this direction. Maybe compartments were necessary very early on. I remind the RNA world hypothesis. So you know the central dogma of biology. DNA is replicated. You have transcription and translation. And so the RNA world idea is that at the beginning, RNA was kind of an autonomous systems with entities that could copy themselves and perform some catalytic activities, thus replacing DNA on proteins. So we know that RNA is a sequence that's a messenger, so it has a code, but it's also a catalyst that was discovered in the 80s. And the type of catalysis that was found was in introns. So in cells, in messenger RNA, you have introns and exons, and introns are RNAs that excite themselves out of an RNA. So it's an RNA that folds and catalyzes its own excision from the RNA. So you have a product where the intron is out by its own action, and then you have the ligated exons. There is a whole family of such objects, and a lot of organisms have group 1 introns, for example, or other introns. Here is an example of the tetrahymena, one of the early studied. But the ribosome is also a catalytic RNA. So catalytic RNAs are called ribozyme. And the ribosome is a ribonucleoprotein protein, which is made of RNA and proteins, but the catalytic activity and all the main activities are performed by the RNA part of the ribosome. So this is a strong argument in favor of the RNA world, meaning that if you look at the molecular hierarchy in the cell, It's kind of a ribosomal RNA that takes the transfer RNA and the messenger RNA and makes proteins. So we are among the people who argue that the RNA polymerase ribozyme did not appear from random sequence and started evolution, but we go along this possible path. We try to build a path where there could have been a gradual acquisition of evolutionary properties with what I described, but here a little bit more precisely, where the RNA oligomers self-assemble. Some self-assemblies have catalytic properties. Some of the ligated RNA are also catalysts and autocatalysts, and at some point you may have processes of ligation. And if you lose the triplet, RNA triplet polymerase ribozymes start to exist, so it's possible to imagine that you don't do polymerization from single nucleotides, but you already use preformed parts. And maybe ultimately you have an object which is a true polymerase in the sense that we understand. So obviously a big focus in the field was to try to find the RNA self-replicase, which would be this polymerase that catalyzes a copy of RNA into RNA using nucleotides. So now this is a bit dated article. In 2013, already since more than 10 years, there are RNA that can copy other RNA as long as themselves, catalyze a copy of RNA as long as themselves, but they cannot copy themselves. They can only copy some particular sequences that basically don't fold or don't fold too much. This year, in the group of Joyce, for example, so there is a lot of progress in this field. There are replicases that can copy other RNA which are ribozymes, so those that are folded, but they can only copy smaller ribozymes. It's already a big step because we can have evolution of RNA catalyzed by RNA, but they cannot copy themselves. Another big problem I will come to is that even if you have such an object, it should be very fidel, and this problem was identified by Eigen very early on. So this is a law where you have the genome length, which is bounded, upper bounded by this number, which is the selectivity of the sequence, meaning this is the log of the replication rate of correct copies divided by the replication rate of erroneous copies, divided by the error rate. So you see that the larger the error rate, the smaller the genome. And this is a law that is pretty much verified in biology, where you see that the genome size is inversely proportional to the error rate. So, I mean, this poses a form of paradox. It has been formulated as paradox in the sense that, okay, if you are longer, you need to make less errors. But to make less errors, somehow you need to be longer because you need to be a more elaborate RNA. So we don't know yet, as I showed before, whether there is an RNA that's fidel enough for its own length. There are RNAs that are fidel enough to copy other smaller RNAs, but we don't know. So it will be a very important finding to find a self-replicase because it would show that such a regime exists. It would not completely, it would not close the problem because it's not clear then how it appears, but there is this problem to solve, and compartments can play an important role here. So the first, not the first fix, but one of the main fix that was proposed for the error threshold was a stochastic corrector model in the 80s, where you have a second level of selection. So here the small dots are copies of the RNAs. We have RNA replicators. And if the correct copies, which are the black ones here, are in sufficient number, the compartment will survive. For some reason, that needs to be made explicit, but the model says, okay, the compartment survives, there is such a function. And if there are too many erroneous copies, like in white, then the compartment dies. If you have this second level of selection, you can improve this bound I showed before. And there is some hope, because I would like to point out the fact that we think a lot about mutations, but when you do the experiment, we'll see what happens is that you you have the RNA polymerase jumps. So your copy is smaller. And the smaller the copy, first, it has a lot of chance to be non-functional. And second, it's faster to copy because it's smaller. So the problem is that this S in general is smaller than 1, meaning that L is 0. So log of S smaller than 1. So you have a big problem because typically if you have such an object, you are going to make one smaller copy and everything is going to disappear immediately. So this is a work that we published a little bit less than 10 years ago now. We did some theory also in these articles later to explicit what was the error threshold in these cases. So if you're interested, I can give you the references. And there is another problem is that, okay, the stochastic corrector model is a model where you have growth and division of cells, protocells, and this in itself is a quite elaborate function. Well, maybe it could have happened, but here we propose something even simpler, which is transient compartmentalization, where objects are compartmentalized, incubated, so you have copies in compartments, and then the survival, there is a selection at the compartment level, but there is no growth and there is no cell division. You just repool everything. So what's really important is that you have this compartmentalized selection step. This is something already well known in ecology, ecological models. And so because it's very more rudimentary, you can imagine more natural phenomena, which are not protocells, but we could lead to a form of transient compartmentalization. Even so, more basic protocells could perform compartmentalization without gross division machinery. The system that was inside was RNA that can be copied here. It's a model system. So we introduced artificially a protein, the Q beta replicase, like in the classical Spiegelman experiment, which can copy RNA. So you have a domain that can be copied here in blue that's necessary for being copied. And then you have a green domain, which is a catalytic domain, which is a model activity because this is a VS ribozyme that cuts RNA, another substrate RNA, and gives rise to fluorescence. So it's a model of a metabolic activity. So here you have a basic object which has two properties which are being replicated and having an activity necessary for selection. And so to study the transient compartments with selection, we encapsulate them in microfluidic droplets. So here you see water in the horizontal axis that is being pinched from below and above by oil. And this is something very easy to make now. You can make thousands of droplets per second. And after, you have a device that performs sorting based on the fluorescence. So this allowed us to impose a form of selection. If you have already done FACS with cells, this is the same type of plot you get. We can have the catalytic activity due to the cleavage of the substrate RNA, which is the orange fluorescence. And then the green fluorescence is a marker of RNA, and which tells us how much RNA there is after replication. And so we have different categories of RNA depending on how much they are replicated or how much they are active. So the initial molecule is both very active and well replicated, and this is, you expect to see it more in the top right corner. You have empty droplets because we put very few RNA per droplets, maybe sometimes less than one. So this is in the bottom left corner. And you have now some droplets which are where the material is well replicated, but there is no activity. And this is what we call the replication parasite. They kind of lost either segments or had mutation in the catalytic part. So here is the result of the experiment. This is a fraction of ribozyme you observe over the whole population of active ribozyme. So if you do the experiment in bulk, you recover the famous result of Spiegelman from the 60s. You have a complete collapse of the activity. So bulk, no droplet. If you do the same experiment in droplets without selection, it takes longer, but ultimately you lose the activity because you start to have some parasites. They invade the droplet, but they only invade the droplet. So it requires a pooling step so that they progressively invade the whole population of droplets. And then if you do selection, what you find is that it's stable until it looked not stable. So all those experiments are not particularly easy. We were a bit stuck at this point, and then okay, what's going to happen here? Is it really going to work? So what's happening is that if you do selection at a lower occupancy per droplet, so it's very important how many molecules you incubate at the beginning, you can repurge the population and recover a higher fraction of ribozyme. But if you do it as it was happening in the experiments, where the number of encapsulated RNA were higher and higher for some normalization reason, then you would lose the active droplets. So this shows that there exist selection regimes in which you can preserve active sequences, despite the fact that replication leads to shorter parasites. And so the selectivity coefficient in bulk is always lower than 1. And the possible genome is of length, but it's not possible. It's of length smaller than 0. So there exists such regime, but why did this happen? I mean, a priori, if you eliminate the droplets that are inactive, it should work. Well, we discovered, and I think that was it was not expected, that for me was the main finding, that there are a new category of parasites. If you do it in bulk, you have very aggressive parasites, which are shortened molecules that appear, and they invade the population, and there is a collapse of functional molecules. But if you do selection in compartments, like in this regime, ultimately you would land on a certain level of coexistence, and there is a coexistence between active molecule and what we call soft parasites, which are erroneous molecules that are replicating at the same speed as the active molecules. So they just lost the activity. And the optimum for those parasites is to the mutant doubling rate here that you see on the y-axis, and the optimum in red is actually to be matched with the wild type, the functional molecule rate. So it's interestingly, this type of selection process selects automatically for molecules that replicate at the same rate as the other molecules, as the functional molecules that preserve selection. Another interesting point is that if you look at the diversity, the bulk selection in blue leads to rapid death. So you see you have a little bump of diversity, which is due just to all the mutants. If you do selection in compartments, you have the same phenomenon, but amplified over time with a larger transient diversity that then collapses. And if you do it with selection, you have a very strong increase, very contrasting increase in diversity. So the take-home message here is that with transient compartments, it is sufficient to preserve function against parasite during replication. Interestingly, you find a new category of parasites, which we call soft, and there is spontaneously a synchronicity that results from selection. And also, you generate diversity at constant length. So this was the first part, first result. I see there are some chat questions. First question, when you attempt to define the question, do you define life? No, we don't define life. Indeed, it's as we all know, it's a difficult exercise. You always find exceptions. So we decided to focus on the question. And so the criteria we have, which are historicity, spontaneity, and similarity with life. So this third criterion is close to your question. We take them and we examine the different articles in literature according to this criteria. Well, my viewpoint is that I could not spend my life defining life because it's an endless question. So I take a very operational definition, and I'm okay with a classical NASA or Joyce definition of a self-sustained chemical system capable of Darwinian evolution. It does not fully define life, obviously, but if I can make such a system, I would be quite happy. It would be a system that would be very different from all the systems we know so far. And another question from Anna. The talk is not finished. Could soft parasite still be beneficial somehow? Yes, absolutely. So that's why I point to these articles from the colleagues like Ryo Mizuchi, Furubayashi, and ESPCI here on the bottom. because they have a system. We stopped those experiments in the lab, but they do a similar experiment with a more convenient system, and they pursue a lot of rounds of evolution, and they do find some RNA that emerge and are useful, are helping the replication. So you have then a lot of phenomena that set up with some feedbacks between the RNA. So this is very interesting. Question from Tanner. Could you comment on what is the minimal diversity at which evolution on synchronicity would be possible? I never thought about this question. 100,000 of variants. Well, there is an obvious answer to this question, which is that synchronicity is very basic in the sense that it comes from the length of the RNA. So we did sequence the RNA and look at them. And so what we see is that we preserve RNA of the same length as the original RNA. So any variant that has the same length, but just some loss of function mutation is okay. So the answer is basically you just need one, but the potential number of them is huge. Like it's 4 to the L, where L is the length of the RNA. Okay, if there is no more question on this part of the moment, I will continue and drink a bit of water. There is, now we looked at template-based replication, but so I showed you this more gradual idea of prebiotic evolution. And there are systems, first, so theoretically, of autocatalytic set, where you have a set of molecules that help to make each member of the set at some point. And this idea, I'm not going to comment on those diagrams, but is that if A helps making B, and B helps making C, and C helps making A, you close the loop, and at some point, you should have a form of amplification. And so Kaufman did a calculation presented in this article where he said, okay, it's very plausible that you have such a phenomenon in the sense that if you have enough molecule at some point, there are enough reactions and you have a form of percolation in the physical sense where you will find a cycle somewhere. And this is going to occur. So there are a lot of caveats in this reasoning, but at least it started the idea. Well, the thing is that they are experimental systems that are not replicas, as I showed before, but are just made of RNA and do a form of autocatalysis. So there are mostly two systems. There is another system I don't show here from a Japanese book, but there is a Joyce ligase, which you see on the right side, where you see this gray part on the bottom. It can catalyze the assembly of the two parts on the top. And so this could occur reciprocally, because you could imagine then that you have substrate on the gray side that are cut in two and assembled by the now full-length black part. And this mutual process from the fragments, 4 fragments, could lead to an amplification of the two molecular system. There is another system, which is the one I show in my studies, which is from the Lehman group, where you have some fragments, they self-assemble, and this object, if it's covalent, is going to help the covalent assembly of the fragments. So it's slightly different because you have much less templating in this system. So interestingly, here we have two systems that do autocatalysis, and we are shown to do so in the lab, and they work in a slightly different manner. But none of them is a polymerase. So it's not very straightforward to imagine. We have to imagine how such an object could evolve. So it's easier to see on a video. It's a video of the system of the Lehman group that was before on the left side. So you have those fragments, and because you have base pair complementarity, they can recognize each other and make a complex. But this complex is non-covalent in the sense that you see here on the top, for example, they are not attached. So this complex is a bit unstable. But if you have a fully covalent complex that's stable and going to appear in purple from the left, it will be able to catalyze the bonding of the dangling ends. So you will end up with two covalent copies of the initial molecule. And you see here, there is no templating in the sense that you don't have several partners. There is not the catalyst, the template, and then things to assemble. You have a complex, and then you have a catalytic object that arrives and attaches everything together and make a second molecule. The question of Henrik, I didn't discover them, but I can come back to it later. Yeah, this is an interesting question, definitely, about peptides and RNA. So here we have to take a step back because we cannot associate directly Darwinian evolution with template-based replication. And so we have to think more generally. And provocatively, I have this little caption of Darwin. He never heard about DNA. So when he formulated evolution by natural selection, he said basically there is variation, reproduction with heredity, and selection. But he did not know how. And so now, I mean, I guess it was such a revelation when we understood how it occurs in cells. It's very hard to get rid of this picture, but that's a little bit the exercise we play to try to imagine other ways to have those properties without having, strictly speaking, the DNA replication or a similar form of replication. So those two systems I showed before, they have been diversified a little bit in this substrate binding region, which can lead to a more collective replication. So this can be discussed a very long time. Is it a form of evolution or not? I think at the moment it's not really, it's a form of evolution in the sense that you do have diversity and reproduction, but there is not in this system a moment where a specific identity different from another one is submitted to selection as a whole. So you are not seeing truly preserved generations submitted to selection. But yeah, this is a possibility. So we wondered whether there could be what would happen if you have some such of collective reproduction. Do the way they interact provide some differences regarding the Darwinian properties? I must say that this study is not completely straightforward to understand in the sense that it's a bit ahead. It's missing one part, which is heredity. I was saying this before. It's just we just made different systems with different interaction and composition and see how they behave. And if such system exists, it would give some rules, some constraint about how evolution should occur given collective autocatalytic sets. So it's not really straightforward when you have a network of objects that interact with specific interactions, because they could be highly cooperative, they could be non-cooperative, like on the left. You could have new species. Imagine evolution is leading to the appearance of a new species that impacts a lot of species or it does not interact with them. And what would happen? Well, if you have a poorly connected network, you certainly have little reproduction. If you have something very, very strongly connected, maybe it's quite robust, but we don't know. If you have something that a new species that arrives but is not connected to the rest, it's going to not cause much change. And maybe it's the same. If you have a species that arrives and connects to everyone, it's not very clear what's going to happen. So this is something we could study experimentally with microfluidics by creating a landscape of different autocatalytic networks and try to explore the behavior of autocatalytic systems as a function of their composition and their interactions. So we inject fragments of the RNA I showed in the video, and this is a video of droplets. The droplets are typically 10 micron wide, so they are picoliters. And you fuse different mixtures of RNA to a big leader droplet, and the big leader droplet here is containing the magnesium and all the buffer for the reaction to occur. And so you create a high diversity of objects like this, and you can use techniques from single-cell sequencing that were developed 10 years ago to really sequence the content of every single droplet. So I'm not going to go into the details, but what you do is that you input some DNA barcodes that are specific to every droplet, and you can then barcode the RNA. All the RNAs in the same droplet are going to have the same DNA barcode, and that you will sequence everything and see who was with whom. And so this allowed us to generate a kind of landscape. So here, each ray is one type of network. And where you have color means that you have those species, one of the 16 species, and the color is telling you how much of it there was. So if I detail, this is how it looks. Here, for example, on this ray, we knew that we have a network with 5 species that interact in this manner, and we could sequence, by sequencing, know that the relative proportion of species were the one you see here. So I'm going to summarize the result. Basically, the result is quite close to a linear model if you simulate the kinetics, where the production rate of each species is proportional to the sum of how much is produced by other species. So it's a collective behavior, and you have a basal rate. And so what we could see is that, well, what was not completely obvious from the start is that you have big differences in how fast they grow, which you see on the panel B. So the yield is the total amount that's made. And here you have a ranking of the different networks. So some produce like 15% of new RNA from substrate, and some almost no. And we also introduced a measurement of how much the relative proportions are perturbed by new species. So here you have a network G, and you have a new species that's called GG. And you can see that the proportions change. So this is a factor of variation. You have a new RNA species arriving, and it's shuffling the relative proportion of the other object. And that we measured basically the difference between the histograms, which is this measure, PGG. And if you rank again by difference, what you see is that you have a lot of type of different situations where you completely shift the composition or you don't at all. One question, would nucleic acid catalytic network look substantially different, more complex, I assume, if nucleic acids were made from more than 4 letters? I cannot answer the question, but I'm going to give you some clue that could help to answer this question. Yeah, for me, it would mostly be a matter of degree. Like we would find the same typical laws, but we would have more room for tuning the interaction, so we could populate the degree of interactions differently. So basically, the law is here. The degree of perturbation, so this is kind of addressing your question, depends on 3 parameters mostly, which have to do with the pre-existing network and the specific interaction with the new species. So the main parameter is the network strength, is the total strength of the links within the pre-existing network that you see in black here. So sigma G, the strength is very strong. Everyone is making everyone and with a lot of intensity. And then 2 parameters have to do with the new species, the novelty. So in the sense that is a new species creating an interaction, so catalyzing a species that was not catalyzed by anyone else before. So for example, here you see that, yeah, everyone was more or less catalyzed already by someone. But if GU would, for example, GA here is not catalyzed by anyone. So if a new species would catalyze it, it would be very novel. And the 3rd parameter is the targeting breadth, is that how many species is targeted by the new species? With how many pre-existing species does it interact? And so the degree of variation depends on this law. It's something that has to do with how strong you are to begin with, how novel is a new species, and how broad is the effect of the new species. And to summarize it in one law that we tested here on those curves, I don't have time to detail here, but strong perturbation. So if you shuffle a lot the composition, you have to have a catalytic innovation, a very novel species that targets sparse network. So this means if your network is very strong, it's very robust. And if you're targeting your new species is novel, but is not only affecting one species, it's not going to have a lot of impact. So what we observe on top of that is that you have kind of a trade-off. if you have a network that grows a lot, so this is here the yield, typically you will rarely see it being perturbed by a new species, which is consistent with what I said before. So this top corner means, okay, if you are a very strongly connected autocatalytic network, you can be perturbed a little, but you can almost never be perturbed a lot. And all the other cases are possible in contrast. So if you think about all that together, you have a trade-off between robustness and variation. So to have a strong perturbation, you must target a very weak network with poorly connected species, but you must also target a lot of them. So you are going to create a lot of connections. So after a strong perturbation, you typically end up with a strong network, which is hard to perturb in turn. And that's what we see on our trajectories. If we look at the cumulative perturbation, if you have big perturbations, they are generally followed by plateaus. So evolution is a bit like this punctuated equilibrium theory of evolution. So on this part, the take-home messages are that you have a large range of possibilities of differential reproduction and variation in autocatalytic network, even with a very limited repertoire of specific interactions. So here we had only 16 species, and there were only 4, so 4 by 4, actually variations in how they interact. So to come back to the question, if we would have more diversified nucleic acid, we would just have a larger repertoire that would populate more continuously this space. And the variation in composition in a collective system are controlled by the strength and the diversity of the specific interactions. And interestingly, we find some trade-offs between growth, variation, and robustness. I don't know where I am with time. I'm going to continue. So now I move to yeah.
Just want to say you have about 15 minutes till our usual stop, but you can keep going as long as you want.
Okay. Great. So there is one question here. Would collective autocatalysis network behave similarly if those were catalytic peptides instead of RNA? Well, frankly, I would tend to say yes, but in the detail, so autocatalytic peptide network, like the one of Gonon Ashkenazi, they are based on partial templating. So the strength, the specificity, I believe, can be higher. The other factor is that in the regime we explored, the kinetics is pretty well explained by such a linear relationship. So if you have a very non-linear system, that may become quite different. But I must say I would not be able to go further. I'm very interested in what comes out of Gonon Ashkenazi experiments in this regard. So yeah, my title is Semi-Permeable Compartments, comprehension of semi-permeability. So before I showed you compartments which are themselves, in the sense that they can be selected, but they do not exchange any material with the outside. And this is a very, very important property. Cells are semi-permeable. Huh? They import stuff that they need, and they export stuff that they don't need or that other cells need. They interact or to do signaling. And like typical membranes, even like vesicles or prebiotic early relevant one, they necessarily are semi-permeable. They would let through some stuff and not some other stuff. So what are the consequences of this semi-permeability on Darwinian properties? So first, I highlight in one slide something we showed in this article. Where we classified the stoichiometry of autocatalytic systems in general. And so we found that you have a typical topology among 5 typical topologies, which is the one on panel B, where you have an object that divides into 2 objects, and then you find a way to recover the first object by 2 different branches, which are pictured by the green arrows here. Well, maybe you can find that in nature, but imagine on panel A, you have a very simple reaction, which is a bimolecular reaction, where has 2 components, the orange and the pink one, that are attached by a covalent bond, could be any molecule. and there is a reaction that leads to separate them into break them, basically. And this reaction in a compartment is very banal and is not doing autocatalysis at all. But imagine you put it in 3 compartments, the same reaction, that are separated by semi-permeable membranes, which are described here as colors. The pink membrane only lets through molecules with a pink part, so pink alone or pink with orange. And the orange one lets through molecules with an orange part, so orange alone, or the combination of both. And so you see that you can recover the scheme on the top, which would allow a form of autocatalysis. And so this is would be a magic of semi-permeability. We have not seen system like this for the moment. So if you want to make a system like this, we would be very happy. But by the fact that you create molecules that are able to import the molecules to go through the membrane, you see that you have an amplification phenomenon from 2 reservoirs of simple molecules, and then you amplify the amount of more complex molecules. And this emerging behavior of autocatalysis is due to the semi-permeability of the compartment. So this shows you that this is a very powerful idea, the semi-permeability. Here, when you look at the membrane, it's a bit like if it was like introducing a new reaction. So now I'm going to explore the potential of semi-permeability in a slightly different context. It's a little bit the same idea. So you have some objects that go traverse the membrane and some that don't. And this, now, instead of implementing autocatalysis by semi-permeability, we directly took an autocatalytic reaction. So this is a very famous autocatalytic reaction called the Formosa reaction, where you have molecules with 2 carbons. And when you have a molecule with 2 carbons, here we call C2, there is a relatively spontaneous reaction that makes a 3-carbon molecule by integrating the C1, which is formaldehyde. And after C1, you can make a C4, so 4-carbon, and the 4-carbon molecule can be broken in twice 2-carbon molecule. So you see here, you start with 2 and you end up with twice 2. So you have an amplification phenomenon by growth and division of the molecule. This is a reaction that does work in the lab. And so what happens if you put it in our case, it's a droplet, so a compartment, such that the small molecules, because they are small, they can quite easily exchange, but the bigger molecules that result from as products of the reaction, they are a bit too big to exchange. I mean, they exchange, but much, much more slowly than the smaller reactions. So we are in a case where it's okay, you can be exchanged between droplets if you have 1 carbon or 2 carbons, but larger, then it becomes really slow. And so the scenario, yeah, you have kind of a phase diagram. Here in color, you have how much you grow. So if you want to grow to double in volume, it's the green line with 2. Sometimes you shrink, and it depends on mostly 2 things. You have to compare how fast you exchange the resource, which was the 1-carbon molecule, the formaldehyde, with how fast is the reaction. So this is the x-axis. This is quite understandable. It's important. Like if you can exchange the resource, but then you're really slow at doing the reaction, maybe it doesn't matter that you exchange the resource. The y-axis is another important parameter, is how fast is the reaction divided how fast you do the reaction. Oh, sorry, I was a bit confusing. So the x-axis is how fast you exchange the resource with how fast you exchange the catalyst. And the y-axis is how fast you react divided by how fast you exchange the catalyst. So you have 3 important timescales, and you have to compare them with each other to find a regime in which an autocatalytic reaction could cause the growth of a compartment, so the import of the resource from one droplet to the other, due to imbalance in growth. I come back to this phenomenon. So what is happening? Imagine you have a droplet where you have a fast reaction, is the red one, and a droplet where you have the slow reaction, is the blue one. The red one is going to consume the formaldehyde, so the blue squares, very rapidly. And so there will be a gradient of concentration of the blue between these droplets, the right one and the left one. and you have a law of diffusion, so Fick's law, the law of Fick, is going to cause a spontaneous flux of the blue to the red droplet, because the blue squares can exchange. But you make larger objects that are trapped in the droplet. And so, because they are trapped, the number of objects is going to increase in the red droplet. And then you have another phenomenon that's called osmosis, which will tend to equalize the total concentration. So as you accumulate more and more products in the red, you will pump the solvents of the blue droplet, and then you will grow. So by this mechanism, you could have an increase in volume that's due to the efficiency of the autocatalytic reaction. And all that is due to autocatalysis and semi-permeability. And so semi-permeability, the properties here are those values. They express how semi-permeable you are. So we did this experiment with droplets. So some droplets are tagged with green. Those are the ones where you have inefficient autocatalysis. And some droplets are tagged with not tagged, they are gray, and you have efficient autocatalysis. And so what you see is that the gray one, they grow at the expense of the green one. You also have the confirmation that there is a form of competition, that a gray one in the middle of other gray ones is smaller. It had less access to neighbors which were poor at catalyzing. So you see a direct effect of the composition of the droplets, which lead to a form of diversification in the growth behavior. On the contrary, if you have some green droplets that's surrounded by a lot of gray droplets, it shrinks a lot. Like it has a lot of very strong competitors. And so it's pretty artificial, but imagine you have, in this configuration, but imagine you have a porous medium or a flow that's causing some shear disturbance on the droplets. Bigger droplets are more fragile. Their surface energy is lesser. So if you submit them to a form of shear, like here in a porous medium, they are going to divide, whereas the small one, you see, they don't divide. So first you have autocatalysis, which is more or less efficient. And when you have efficient autocatalysis inside you, you grow. And when you grow and you are submitted to shear due to hydrodynamics, you divide. So you have kind of a way by which, by purely a physical-chemical mechanism, you have a form of fitness. And now you have this new population, and we show that you can re-inject it and then resubmit it to the same type of competition of growth. And what we see is that this growth at the second round is partly heritable. So if you grew lots at the first round, you will tend to grow more at the second as compared to those that did not grow. Because you are bigger, so your interface to pump the resources of your neighbor is bigger, so the rich get richer, unfortunately, and you are going to grow faster because you were bigger at the beginning. And so this is a beginning, this is a rudimentary way of having heritability, which is one of necessary for Darwinian evolution. I would like to highlight something that is hidden in a supplementary figure, that imagine you have not a perfect porous medium, but a violent ocean that's dividing the compartments. And so the division probability depends more or less stringently on the droplet volume. So what we did in the experiment is a blue curve where you have really a threshold. But imagine if you would have a very turbulent flow, it would be more the red curve. And so interestingly, when you have new competition, you would have growth and division. You really need the threshold division volume to be very precise. But if you have competition, you really enhance those contrasts. And so you have a much broader range. Here now it's a log scale over which you do have a relative fitness that emerges. It should be competition, I'm sorry, due to competition. Question, in the droplet growth experiment, what is the food source? So the food source is in every droplet. It's just that some of them are able to make something out of it rapidly and some are not. So those that are not good at transforming it into larger sugars, basically, they lose it at the expense of their neighbors who are very efficient. Dieter, stating that the formal reaction can be performed in the lab actually require to state that the boundary, the pH, the pH is very high. Yes, it's 14, 13, 14. Fast kinetics, yes. Well, you're right, it should be mentioned. Well, yeah, Dieter, I mean, other reactions, this is a bit a problem of origin of life. For me, if you know other reactions that we could use, we would be extremely happy. But your question is very meaningful, obviously. To get that to actually work, in our case, we needed to find a base, which was a super base that allows to maintain the pH and can exchange between droplets. So what I hide in this description is that you have the formose reaction and the carbon molecules and the sugar and the formaldehyde, but you have all the rest that allows the reaction to run. And you should make sure that the minerals and all the rest are also present in every compartment. So this is an additional constraint on the semi-permeable properties of the compartment. They should allow all the buffer, let's say, to enter the reaction. Yeah, I'm basically done. I mean, this is a kind of a take-home message, a summary highlighting the role of compartments for replicators on the way to make maybe an artificial cell, or at least in my case, I'm aiming for a Darwinian system, whatever it is. First, I showed you that compartmentalized selection avoids error propagation. And this is not just a small qualitative effect. You can go from the regime of, as I told you, the copies typically they are shorter, and so the error threshold is genome of length 0. But with compartment, you can go to genome of length more than 0. Otherwise, you have to postulate that you have a high fidelity replicase and that this replicase never jumps, so never makes smaller objects. And I should add to that it's never discussed in the literature, but there is a magic thinking here that an object appears from random RNA, and as we know, random RNAs are distributed, or random polymers in general, exponentially. So you are almost guaranteed to have any, sorry to say that, pardon my French, but any crap around, any possible parasites is typically around for a generalistic replicase. So it's really a very big problem of emergence. Now, the second point is that as compartments, they are necessary if you think about collective autocatalytic sets, because you need a unit of selection. You need a physical entity that makes it a Darwinian unit somehow. Otherwise, if everything is mixed, like in the experiments that were done by Joyce and Lehmann diversifying the interactions, you have a collective, you have an interesting phenomenon, but you do not have a Darwinian unit per se. Now, the last 2 points are about semi-permeability. So I showed you that with semi-permeability, you could have new ways to implement interesting feedbacks, and in particular, autocatalysis. And this is just a theoretical thought for the moment. It would be very interesting to see it realized in the real life. And finally, semi-permeable compartments couple growth to autocatalysis, ultimately allowing selection if you add another ingredient, which is that division caused by some forces, depends on size. And this was an idea already proposed before by Jack Szostak in his vesicle RNA system. Yeah, that's it.
I think there's another question, 2 more questions in chat.
Okay, thank you very much, Mohamed. I come back to the earlier question on the peptides. Yes, I mean, so the question of Henrik was, did you consider scenarios with RNA co-evolved with small peptides, with peptides aiding RNA activity and catalyzing an RNA catalyzing peptide synthesis? Would the system behave different than pure RNA? Yeah, absolutely. I mean, my dream is to study that next. I think there are some possibilities now to have some form of protoribosome. It's a very, very challenging and very, very central question. Like many people, we are also studying coacervates, which are mixture of RNA and positively charged peptides. In this case, is Tommaso Francia, who is here. And already in a very basic sense, it's interesting to see that you can make spontaneously formed compartments just by making this mixture. So even on a very, very basic level, it's helpful. Regarding all what I showed, would be a way, simple way to make compartments that could help the emergence of evolution. But all your questions are very fascinating. I hope I can answer you in 10 minutes. Yeah. So Mohammed said, yeah, thank you very much, Mohammed. Can you speak how important enzyme catalyzing reaction is versus spontaneous chemical reactions? How likely is it for minimal enzymes, RNA catalysts, to spontaneously appear? The second experiment shows that you can get evolution from spontaneous chemical reactions, and I'm curious how far that can go without enzyme and catalyst. Extremely interesting question. So we have a PhD student in the lab, Pranit Nandan. I'm first studying this theoretically because this question fascinates me a lot too. Can you really have a diversity of autocatalytic system? And either way, you could kind of transit from one to the other. I wanted to study that in a more realistic setting than Kaufman, so with small molecules and spontaneous reaction, as you mentioned. So our work on the classification of autocatalysis allowed us to start to tackle this question. Some other groups are starting to do that. It's a very challenging. So we are generating chemistries artificially. And the problem is that in chemistry, it's very hard to predict activation barriers. So we have to find a way of abstraction where there is valency, the atoms are described, and some realistic aspect. But we also have to randomize some part of the network, like Kaufman, in the limit of what chemists can predict and can know. And so what we are doing is we are exploring whether it's plausible that there is autocatalytic system in this. Now, engaging in this question, what I realize is that my first question is, can the system evolve? And then you are wondering, is it plausible that there is autocatalysis other than the Formose reaction? Is the Formose reaction I showed super weird or not? We don't really know. And then now I'm just wondering, okay, why do organic molecules grow or not due to spontaneous reactions? So I'm reducing my ambitions progressively, but also maybe this type of program can help us to interpret. Now people are doing these more messy chemistry experiments. So I have colleagues like Grégoire Danger in Marseille, or I guess there are people in Munich, making complex mixture and trying to see what comes out of it. Maybe longer molecules, how, and maybe autocatalytic system. So this is really a new question, I would say, that people are starting to tackle. Yeah, but this is something we do now. And Dieter asked, the replication experiment is the first part, rely on base-by-base replication, absolutely. I would expect that things change a lot if replication is performed by spontaneous templated ligation. Yes, without the need to Joyce ribozyme, for example. Would that templated ligation not create an autocatalytic reaction directly by the hybridization of the RNA strands without having to wait for ribozyme function, which only kick for quite large sequence? Okay, well, maybe you have your own idea about this. What we are doing, we are trying to do that now experimentally. To have indeed templated ligation of substrates. Well, we are a bit back, it's a complex question. So we are a bit back to the Joyce case somehow. But if any system can undergo this, which one would come out? But you're probably aware of the experiment of our colleagues Mizushi and NGHE that they published last year. So they mixed random RNA. I'm really fascinated by this experiment. They mixed random RNA of length 10, I think. And then they saw that there were emergence of some species that would tend to catalyze the appearance of each other from fragments. So for me, it's very exciting that this is happening. And it's also exciting that those objects kind of look like a primordial version of the Joyce ligase. I don't know what's your opinion, Dieter. You are also engaging there, no?
Yeah, longer discussion, but it's a good starting point, I think. Yeah.
At least for me, the problem I try to tackle now, because you always reduce your ambitions when you identify sub-problems, what are the conditions for multiple turnover? I think that you published on that recently. That's really the key thing for me in the RNA world. Yeah, it's historically the problem.
Thank you. I think you basically gave a second talk answering questions. That was awesome. Thank you.
Thank you very much.
I was really happy that someone asked about the multiple different non-canonical bases in RNA, because that's something we're starting to look into ourselves too, and I would be really curious to see how some sort of a catalytic networks out of non-canonical nucleic acids behave.
Do you expect stronger interactions, or?
I honestly don't have any specific expectations. I would probably expect, so some of the bases we're using have a little stronger base pairings, so maybe the reactions would be slower. Maybe there would be more temperature dependence or temperature variance. I don't know how that would affect the dynamics of the networks. That's something that I'm really curious to see.
But maybe you could probably broaden the range of conditions under which something interesting is happening, which is a very important point. When you try to go to this naturalization question, like the historicity of the environment.
Yeah.
Or even practically in the lab, it's always good to have more possibilities.
Definitely. All right, thank you so much. Thanks so much for giving a fantastic talk, and thank you everyone else for a great discussion.
Thank you. Thanks a lot for the invitation, and to the audience for all the questions. That was really very nice, very pleasant for me.
And have a great rest of your Monday.
Yes, biking under the rain now.
Oh, well, hope you still enjoy it.
Right.
Bye.